Okay, so now that we have done an overview of biological membranes, we are ready to learn more about membrane structure, which will be the topic for this portion of the lecture. So to start off with, we are going to focus first on the lipid bilayer, right? Because that's the main component of all biological membranes. So just a reminder, the cell membranes are packed with these phospholipids, which contain a hydrophilic head along with hydrophobic tails. The two fatty acid tails are joined together with a glycerol molecule, and then they have a polar head group that contains phosphates, and then along with that some other polar molecule as well in many cases. Because of this, they are what we call amphipathic molecules. So they have the hydrophilic head and hydrophobic tail. They have both hydrophilic and hydrophobic components. The biological membranes are made of these lipid bilayers. So if you remember from last time, we talked about how if you put these lipid molecules into an aqueous environment, they will try to get themselves oriented in a way so that there is minimal interaction with that aqueous environment for the hydrocarbon tails. And they do that by making a bilayer where the phosphate heads or the polar heads are towards the outside and the tails are facing towards the inside, facing each other, and thus excluding the water molecules from that environment. Now these fatty acid chains that are present in your phospholipids can be both saturated as well as unsaturated. Obviously, if they are saturated, they're going to be more tightly packed. And if they are unsaturated, just like we talked about with triacyl glycerols, they will have kinks or bends due to the presence of double bonds. And those double bonds are going to then kind of increase the volume of those molecules so that they are not as tightly packed inside the space. Now, in addition to these hydrocarbon tails and to these phospholipids, there are other components of the lipid membrane or the biological membranes that are also amphipathic as well, right? So there are other types of lipids in there as well that are amphipathic. A big group of them are cholesterols or sterols. You will also see glycolipids in that membrane, where it is not a phosphate group that is attached to the glycerol, but instead sugar molecules that are attached to the glycerol on the hydrophilic heads. In either case, you will still have some component, some oxygen, some ionic component in those structures to provide that hydrophilic portion of the molecule within the membrane along with that big hydrocarbon tail or whatever it is that is keeping the hydrophobic area together. Now, cholesterol is a specific type of hydrophobic fat molecule in the sense that it is not exactly a chain in the typical sense like we're used to with all the other hydrocarbon fatty acid chains. Instead, it is a ring structure, and it's present in many of our cell membranes. It has important functions in there, but it is present also floating around inside our bloodstream all over the body, as it provides important components to produce hormones of various nature inside our body. Now, when we are looking at how these molecules are going to interact inside the normal environment of a cell, well, remember majority of the cell's environment is hydrophilic, right? Or not hydrophilic, but aqueous. So it contains water, right? 60 to 80% of our cell component is just water. So both hydrophilic and hydrophobic compounds will have to interact with water in some way, shape, or form. If it is a hydrophilic compound, that will obviously have the added advantage because it will have either polar areas, so partial positive, partial negative groups, or it will be ionic in some way so that it can interact more directly with the water molecules so that it will be making hydrogen bonds between its own polar groups and the groups in water molecules. On the other hand, if you are dealing with a pure hydrophobic molecule, you are going to have conditions where the molecules will almost make a cage around it using their own hydrogen bonding capacity, so that they minimize the interaction with that molecule that is inside, right? That is hydrophobic. So you can see that right here indicated by these lines. These are hydrogen bonds that have been created with the water molecules themselves so that they can create almost like a cage around this hydrophobic molecule that is there. Now, all these properties are extremely important in the way the lipid membranes are going to be created and then maintained. How they maintain their integrity throughout the time that they are there for the cell. So once again, if you look at this diagram, you can see that you will have the aqueous environment both outside and inside the cell, and those are the areas that the hydrophilic heads are going to be interacting with, and then your fatty acid chains are going to be facing each other inside, excluding that aqueous environment away from itself. Again, these are two main types of amphipathic molecules that you may see inside your lipid membrane, the biological membranes. You have the triacyl glycerols in various ways, right? Those are your storage molecules. And then you may have the typical fatty acid chains with the glycerol molecule with the phosphate, and then some polar group like an amine or a choline molecule attached to it. So now let's go back and think about how the molecules are keeping themselves in the right condition, right, in the right environment. So you have your phosphate heads, the phospholipids, hydrophilic heads that are facing outside towards the aqueous environment. You have the lipid portion, the fatty acid chains, the hydrophobic regions facing each other, excluding the water environment. However, if you were just to create a simple flat sheet out of it, you still are interacting. Some of the sides, all the sides would still need to interact with water molecules or aqueous environment. And that's not going to be very energetically favorable. So to minimize that requirement and minimize that instability, these structures will spontaneously form sealed compartments. They don't have to be perfectly round, and they rarely are perfectly round, but they have to be sealed. And by creating a sealed environment, what you end up with is just the hydrophobic tails inside and just the phospholipid or the hydrophilic heads towards the outside. And this type of sealed compartment is going to shield the hydrophobic tails completely from water molecules and will be energetically favorable. That is how all the compartments within the cell are made. That is how our cell membranes are maintaining their integrity as well. This can be mimicked in a very simple experiment. If you were to put some pure phospholipids or even just oil in a glass of water and stir it up, they will automatically form these droplets or micelles where the fat molecules will spontaneously create these enclosed structures and organize themselves into these kind of liposomes or spherical structures so that the fatty acid chains are away, excluding the water from their environment. And this would be similar to what you would see inside the cell as well. Now, that's all great. Now you have a very clean system. You have the lipid bilayer, and it's in an energetically favorable condition. However, that doesn't mean that they're static, right? That doesn't mean that they don't move and these individual molecules are going to be locked in place. No, not at all. They actually have a lot of movement going on constantly. Most of this movement is going to be lateral diffusion. So you will see movement of these lipid molecules all throughout the cell, all over the cell membrane area. You will also see them rotating on their axes, or moving just the tails, flexing their tails as well, the fatty acid tails as well. On very rare occasions, you do see flip-flops of these molecules from the luminal side to the cytosolic side, or vice versa. However, that flip-flop will require the aid of enzymes. That does require energy and the correct orientation to be present in order to happen. And there are very specific conditions under which this would happen. So why doesn't that happen? Well, there are multiple reasons for that. So first of all, how do we maintain the fluidity of our lipid bilayer, right? So that's the first question. Why is it that molecules can move around and do all the stuff that they can? So the main way that we maintain the fluidity of our membranes is through two main properties of the fatty acid chains. One is the length of the hydrocarbon chain themselves. That length can vary from 14 to 24 carbon atoms. Usually it goes between 16 to 20, 18 to 20, something like that. Shorter chains are going to be a lot more fluid, will have a lot more freedom of movement in many ways than longer tails. The longer they get, the less mobile they are. The second thing that is going to affect the stability of the membrane and its fluidity, how easily these molecules can move, would be the number of double bonds within the chain. Typically, the more double bonds are there, meaning the more unsaturated fatty acid chains are there, the more fluid the membrane can be. Because if you remember, as they make these double bonds, they become a little bit more bulkier, they take up more space, which means that you can't pack them quite as tight next to each other. And that allows for some empty space, some wiggle room, and that allows for higher fluidity. So if I think I have that on the next. If I wanted to recap, this is how you would look at it. If you have short, unsaturated hydrocarbon chains, so these are shorter chains, and they have double bonds, so they have kinks and bends, and they're a little bit more bulky. That would lead to a higher permeability in the resulting membrane and more fluidity in general. These will move around a lot more within their structure. On the other hand, if you have lipid bilayer that has long and saturated hydrocarbon tails, you will reduce the permeability and fluidity of that membrane, because they are going to be tighter packed, right? They'll be more tightly packed, there'll be less gaps within them, so less molecules and smaller molecules would be the ones going through, and the molecules themselves won't be able to move around quite as much as the short unsaturated hydrocarbon tails. Now, that doesn't mean that all short tails are going to be unsaturated and all long are going to be saturated. Quite the opposite. You would see a mix of these depending upon the needs of the cell and the needs of its environment, where these membranes are getting made and what they are going to be doing. Now, once you have that first layer of control, right, with the length of the hydrocarbon tails and the amount of saturated versus unsaturated fatty acid chains, there is a second level of control as well, and this is the existence of cholesterol inside those lipid membranes. The cholesterol molecules, if you remember, are these ring structures that makes them short and rigid, so they are not as easily movable, right? They are going to be a lot more locked into their own little shape. They are used to fill in the gaps that are created by unsaturated fatty acid chains, and they allow us to they allow the cell to kind of play around with how fluid it needs to be depending upon its environmental needs. So cholesterol, when it is packed into a certain region, will make that area kind of locked into its own place. So maybe overall the membrane is very fluid and does not move a lot, or moves, those molecules are moving constantly, but maybe there are small regions within the membrane that are kind of locked in their place. those areas are actually called lipid rafts. Those are areas that are going to be super saturated with the cholesterol, not only cholesterol, but they're also going to be super saturated with transport proteins, helper proteins, and those are kind of signaling hubs. They're like your signal towers, right? They are the ones where signals come in, where molecules get shuffled through to interact with the environment, to create responses, all kinds of things. So that is something that we are going to be talking more about next week, or not next, yeah, a little bit next week, but also in the next lecture. However, that is something that you can know. Sorry. Okay, so cholesterol in their rigidity and the small size can fill up some of these gaps and increase the, how rigid and how permeable, to decrease the permeability and make an area of the membrane more rigid, allowing for more controlled environment in that space. Now, when we look at the membranes, we now, have been talking about for quite some time about how it is a lipid bilayer. Well, not all layers are the same in this, not the both layers of the membrane are not exactly the same. They are actually quite different in their composition of the type of lipids that make up that side. Our extracellular space has very different environment and very different requirement for these phospholipid molecules. So not only do the membranes change in composition and in their character by organelles and by their general space, but each side of the membrane is different. They are according to its needs. So the extracellular space, the lipids in that environment are going to be the ones that are interacting with other cells, with extracellular matrix. They are going to be kind of the first line of interaction for that cell with its environment. And those ones will usually have the glycolipids, so the ones with the sugars attached to it, that aid in cell-cell recognition and cell matrix interactions. In addition, they will also be rich in these sphingomyelins and choline molecules that are attached to the phospholipids. So they will be very specific types of lipids that will be found facing the extracellular space. On the other hand, the ones that are towards the inside of the cell, the ones that are facing the cytosol, are going to be more rich in the phosphate attached to serine or amine group, because those are the ones that are going to be creating the signaling cascades after they receive the signal from the outside. So the inner environment of the membrane is going to be very different, the one that faces the cytosol compared to the one that faces the extracellular space. This is also why it's very rare for that flip-flop to occur. If you remember, I talked about how the membrane fluidity allows these lipid molecules, these phospholipids, to move laterally, to rotate, to flex, but very rarely do they flip-flop. And the main reason for that is right here. This is it, because the membranes are asymmetrical. Now, when they're made, they start off getting built inside the ER. And so in the ER, when they're built, they're just kind of getting randomly put as a lipid bilayer. However, as this is happening, as this process is happening, as the bilayer is getting made, there will be specific enzymes called flipases that will unscramble them, so to speak, right? They will kind of unscramble them by moving the ones that need to be on the cytosolic side on that side and flipping the ones that are going to be on the extracellular space towards the luminal side. And so that allows for things to start happening, for it to start getting fixed. So this process kind of happens as the membranes are getting built, and then as they're getting moved from that ER space to the Golgi, they will go ahead and they will already be in the correct kind of orientation by that point, right? So you will have the luminal space looking very similar to what it's going to look like when it gets outside the cell, and the cytosolic space facing outside, just like it would be interacting with itself. Now, every time it moves, it kind of maintains that position. Occasionally, if one of the molecules is flipped, the flippase would transfer it again in the right orientation. Okay? So the membranes, like I said, maintain this orientation once it is made, once it is created throughout the transfer. So as they go from the Golgi into a transport vesicle, and then finally merge into the plasma membrane, that orientation will always be maintained. Okay, so they are built in them, they are started in the ER, they get oriented appropriately as they're going into the Golgi apparatus. Once they're there, now they're in their correct orientation, and they're going to maintain it all throughout their transport until they reach their final destination. Okay, so looking at just the lipid portion of your lipid bilayer of the biological membranes, the membranes lipids form spontaneously in bilayers in the water. They are flexible, they are fluid in their two-dimensional space. Their fluidity is dependent upon their composition. So that composition includes the length of the hydrocarbon chains. It depends on the number of double bonds, how saturated or unsaturated these fatty acid chains are. And finally, it depends on the number of cholesterol molecules that are embedded within the membrane. The membranes are assembled in the ER. They start off in the ER quite randomly, getting placed into the bilayer. And then with the help of flippases and other enzymes, certain phospholipids are flipped in their right orientation so that the luminal side will mimic the extracellular space, and the cytosolic side would be what the cells or the cytosolic portion of your cell membrane. Now, now that we've talked about lipids, let's next talk about the membrane proteins that are an integral part of properly functioning biological membranes. Before we can talk about that, we're going to just kind of do an overview of general protein structures. We are going to talk a lot about protein structure and function next week, but this week we are just going to, kind of skim the surface of this structure. So proteins are formed through a chain of amino acids in a particular order, as organized by the, in response to your nucleotide code. Now, that is what we call the primary protein structure. It's just the sequence of the amino acids in the correct order. Now, these amino acids would have different properties depending upon their R groups. So some are going to be acidic, some are going to be basic, some are going to be, will have charge groups. Some of them are going to be hydrophobic. Some will be polar, non-polar, right? And because of that, they can't remain in this long chain because the environment is not going to be favorable for it. So they would start to form local folding patterns to accommodate for those variable conditions that they need in order to be in an energetically favorable condition. The two most common forms that they take in that initial secondary structure are alpha helices and beta pleated sheets, which is where they kind of, the hydrophobic areas of the protein chains are going to face each other again and create a helix-like structure, while the outer portions would contain the amino acids that are hydrophilic. Now, once those internal initial structures are made, in the tertiary structure, the entire ribbon gets folded up in a form that is going to be the most energetically favorable one for that particular protein. This is the final active form of many proteins. For some proteins, they do not function alone, but require a secondary partner, either a dimer of itself or another protein, and those are reflected in the quaternary protein structures. where more than one amino acid chain comes together to make the final active protein form. Now, in plasma proteins, you're going to find a host of different proteins that are doing work, important work, in the membrane space. You will have 4 different categories of these proteins. Some proteins are what we call transporters or channel proteins. The job of these proteins is to take molecules from the inside to outside or outside to inside, right? So they act as channels for molecules that are moving through the membrane into the cell or going out of the cell. They act as transporters to actively or passively pick up molecules and bring them inside the cell or take them outside the cell. They are also acting as anchors that help keep the membrane anchored to the internal structure of the cell, or that keep the cell anchored to the outside extracellular matrix or another cell through interactions with them. Then the third category of proteins are receptors that bind to signaling molecules or small molecules and elicit an action response inside the cell in response to whatever signal is bound to it in the extracellular space. And finally, you have enzymes who actually catalyze reactions within the cell in response to environmental cues. So those are going to be your 4 main categories of proteins and the kind of functions that they are going to perform while they are in the membrane space. Now, how do they do this? Well, they do that by obviously being part of the cell membrane, the biological membrane. However, they don't always span the entire membrane space, and they don't always even interact with both sides of the membrane. Some of them will, of course, be transmembrane proteins. So these are proteins where you will have a region that is hydrophobic, that is embedded within the membrane, and you will have areas that are hydrophilic, that are interacting either with the outside environment or with the internal space of the cell. These can have a single pass protein, so one time just passing through the membrane, or they may be passing multiple times and have multiple areas of interactions between the outside and the inside of the cell. Another way would be for a protein to have many areas that can be joined together to create almost like a pore or a little tunnel for molecules to be transported through it. All these are what we call transmembrane protein. Another type of protein interaction with the lipid bilayer is where it doesn't span both sides, but only interacts with either the inside or the outside environment. For proteins that are interacting with the inside of the cell, these are integral membrane proteins, all of these, because they're bound within the membrane. You can have proteins that are directly integrating into the lipid bilayer, into the membrane space on the cytosolic side because of its own structural orientation through a hydrophobic region, as you see in this case. You also have proteins that don't have any hydrophobic regions, but instead are anchored into the membrane through a fatty acid chain itself or a lipid molecule itself. This can also happen on the extracellular space side, where you may have a fatty acid chain that has that phosphate group attached to a long link of sugar molecules or glycosylated. This is a glycolipid that is then interacting with a protein that is similarly modified. So the protein itself is modified, it has phosphates or sugar molecules, and the fat molecule that it is attaching to is modified to contain these sugar molecules or fat molecules or phosphate groups. And that interaction is what keeps them linked together. So proteins that are only interacting with one side of the lipid membrane are called monolayer associated, since they're only associating with one side of the membrane. Proteins that are interacting through a lipid interaction are called lipid-linked interaction. And then some of them are going to be never ever associated directly with the membrane, but will interact with proteins that are integrated into the membrane. And that's how they are working. It could be both within the cell or outside the cells. And these proteins are what you would call peripheral membrane proteins. They are not integrated into the membrane itself, but they are essential for the proper work of those membrane proteins that are integrated into the membrane by associating with them and then eliciting a response. So these are protein attached interactions. Okay. So these proteins, these polypeptides, usually will cross the membrane as either, and when they are integrated into the membrane space. So these transmembrane proteins, when they are actually interacting with the lipid bilayer, they are usually going to do that either through alpha helices or beta pleated sheets. Most of the time, they actually cross as an alpha helices. In an alpha helices, you have hydrogen bondings on the amino acid residues that are towards the inside of the helix, and those would be hydrophilic amino acid residues. But on the outer edge, as they're rotating with these presence of these hydrogen bonds, the amino acid residues that are towards the outside are going to be hydrophobic. And those hydrophobic amino acid residues are what's keeping it integrated into that space. So this would be an example of a polypeptide chain. If you were to look at it, if you were to spread those sequences out, you would see that it would be a combination of hydrophilic, a couple of hydrophilic followed by hydrophobic, a couple of hydrophilic followed by hydrophobic, something like that, where there is a pattern of hydrophilic and hydrophobic residues to create that alpha helix structure. Both the cytosolic as well as the extracellular domain in this case is largely going to be a hydrophilic domain. So here is another example of how you can look at that and look at how they're going to be interacting with their environment. So here you have amino acids. You have a few amino acids that are linked together, and the amino acids have their amine group. They have the carboxyl groups that are already charged. They have negative and positive partial charges because of their own chemistry. And then you have these residues that are hydrophobic in nature that are going to then stick towards the outside of the helix to create that alpha helix structure. These are used many times. These are one way that you can create a pore-like structure or a transporter inside the membrane space. So here is an example where you have a lipid bilayer, and in this particular case, you have multiple alpha helices in this particular protein structure that span up and down with small interconnected hydrophilic side chains that allow for that structure to be formed in just that particular way. And this allows for a space that can be a hydrophilic pore that molecules can go through. Another way that you can have formation of pores within the structure of the lipid membrane is through beta sheets. These beta sheets will have a lot more hydrophobic regions involved in them, and these will allow for movement of molecules that are more hydrophobic in nature as well. But the pores that involve alpha helices usually are more hydrophilic in nature and will allow for more hydrophilic molecules to pass through. Now, when we are looking or studying membranes, we have to somehow find a way to break them apart and isolate them. And we do that by using detergents, just like, grease is cut by dawn. So we use detergents that can solubilize the membranes and separate them out, the individual molecules out for us, so that we can purify them and then look at them. Now, there are multiple types of detergents that can be used. You can use something that is ionic, like SDS, in which case every single protein that you extract after through this procedure is going to have the same charge as that detergent, since it is ionic in itself, and that would be a negative charge in this case. You could also use something that is non-ionic or a zwitterionic detergent, like Triton X, so that you don't change the actual ionic nature, the nature of the protein itself, and you maintain its integrity a little bit better. And in those cases, you can then use those proteins for a lot more sophisticated assays. For example, you can run enzyme analyses with them. So just to review the second portion of our lecture today, the membrane proteins associate with the lipid bilayers in many different ways. They can be transmembrane proteins. They can be integrated to just one side of the plasma membrane. They could be linked through a lipid molecule or through glycosylation sites, right? Or they could be linked with proteins that are part of the membrane but don't directly associate with the membrane themselves. The polypeptide chains will usually cross the lipid bilayer as an alpha helix, and that allows them to create a pore-like structure many times, or a structure that can interact with more hydrophilic molecules. You can also have beta sheets that can create another type of a pore that will allow for more hydrophobic and hydrophilic molecules to be gone through. Now, membrane proteins can be solubilized with detergents, depending on the needs of our experimental design or what it is that we are looking for. We choose the type of detergents to use to solubilize these membranes. We know the complete structure of relatively few membrane proteins actually, just because of the difficulty in isolating these proteins while maintaining their structure completely intact. The plasma membrane is enforced by an underlying cell cortex, which is something we're going to be talking about next time, which has essentially, it's a structural system similar to how the inside of the cell is a structure is maintained through actin and microtubules. This plasma membrane structure is also maintained in its integrity through a cell cortex. This can also be used to restrict the movement of some of the plasma proteins as well as some of the lipids from going all over the place, and maintain their movement within a certain area of the lipid membrane. In addition, the cell surface is coated with a carbohydrate or sugar molecules, in many cases either attached to the lipid membranes molecules themselves, the phospholipids themselves, as we talked about in the case of glycolipids, or to the proteins that are embedded within that structure. So we're going to stop here, and we'll pick up from here in the next lecture.